Great. All right. Well, we're going to get ourselves started. I know there's more folks coming in. So thanks everyone for joining. Really excited to be introducing this topic, our speaker and our guests today. So we'll be hearing from the folks that are behind the Brazilian Reproducibility Initiative, Alavo, a associate professor at the Federal University of Rio de Janeiro, will be speaking to us, and then joined with his collaborators, Clarissa and Mariana, afterwards for some Q&A. So again, you'll be able to have a chance to ask your questions after we get to hear from them. I just want to phrase before we get in here my perspective on this project, just really briefly. I'm really excited to get to this point. It was back in 2018 that Alavo reached out, actually coming to the Center for Open Science in the US in Charlottesville and discussing this project way early when he was conceiving of it with his group. And it's amazing to see the work that's occurred over all those years. And I know how hard it is. It's really hard to be able to pull projects off like this. It requires a lot of work from not just the three individuals you see on your screen, but many other people that contributed to this work over the many years that this project unfolded. And we'll be able to hear about that. I also really am excited to have the community engage with these results. I think the fact that you focused on not just biomedical research, but in the Brazilian context, that's really unique. I think that adds a really great contribution, especially in terms of how we as a community can use these results to kind of continue to improve how we make the most of scientific investment and scientific results. So with that, I'm going to pass the screen over to you, Alavo, and really looking forward to this.
Thank you, Tim. I'll try to share my screen here. Hopefully this should work. Do you see me? Yeah. So yeah, thank you very much. I mean, it's a great pleasure for us to be here, along with people who have really supported the project since its very inception. The first time I talked with Tim was via Skype. It was like the pre-Zoom era. That's a long time ago. And so the newsletter from COS actually said new topics, extra voices, but my first point is like, we're far from new here. I did find a picture of me in Charlottesville in 2018 when I got a Center for Open Science shirt, and it definitely looked different. My hair has a different color, so we've been around for a long time. We're not new, really, and hopefully parents in the audience might get the Dr. Scum from Dogman reference here. But it just took a while, right? So this was the original schedule we presented when I was in Charlottesville, and it said would be finished in 2021. Our first results actually came out a couple months ago in a preprint. So it took a while longer than expected. We did have a pandemic in the middle, which did not help. But yeah, this takes work. And I'll be here to show this work. But we've been around for a while. So the initiative is a multicenter effort by Brazilian researchers to estimate the replicability of particular kinds of experiments in Brazilian biomedical science. We've been covered by a lot of venues along the way. As far as I can see, I think we may have stolen the largest replication project in lab biology from COS, both in terms of numbers of labs and included studies. I do think the reproducibility project cancer biology still has more experiments than we have, so we're kind of like running head to head with them here. But we're the first reproducibility project in any field, I think, to actually look at the issue from a national perspective. It may look funny. I mean, you'd think science is universal and biomedical science is definitely not unique to Brazil, but I do think it's useful for its purpose, right? I think in terms of science policy and what can be done in terms of promoting reproducibility, I think that is mostly in charge of funders and institutions which are local in scope. And I think really putting the perspective in the Brazilian context can help raise debate about this issue in the country, which was really our intention all along. We are a crowdsourced project, so we put up 4 open calls, first one in 2018 for participation, advertised via social media, lectures, a lot of outreach around the country. We did get almost 100 labs to sign up as interested in the project. That's like 20 Brazilian states, it's most of the territory. This is pretty remarkable. We never expected such interest. 75 of them joined the initiative at some point. 56 contributed at least one experiment to the final sample. So these are the ones that remained until the end. And our initial goal is to replicate 60 individual experiments from Brazilian articles using some common methods. We picked methods in which we had a lot of labs working. This was, I mean, kind of like our orienting heuristic here. So we ended up picking the MTT assay, which is a cell viability assay in culture. You see whether cells are able to reduce the MTT compound. RT-PCR, which is a very standard technique to measure gene expression of particular genes in tissue or culture, and the elevated plasmase, which is an anxiety test in rodents. So any experiment within a Brazilian article using these methods and using cell lines, rats, or mice as biological models were eligible to be selected. We did the selection based on a 20-year random sample of articles in life sciences journals with at least half of the authors, including the corresponding one based in Brazil. We did full text mining in these PDFs to look for experiments with these methods that were important enough to be mentioned in the abstract. And of course, we selected these and ran them through our community lab of labs to see if we had at least three labs that were able to do this experiment. So it's kind of like a quasi-random sample of the Brazilian literature, which is enriched in simple experiments that we could do and cheap experiments that we could pay for. Each experiment got sent for application to 3 different labs. The labs were blind to the original source of the experiment and its results. They only got the methods, which extracted from the article. And we calculated sample size to provide 95% power for each individual replication to detect the original relative difference, expressed as ratio of means. As other replication projects, we teamed up with Anna Drebber, Thomas Pfeiffer, and other people to run prediction surveys in markets before the project started. We had 70 researchers predicting the probability of successful replication for each individual experiment, the expected effect size, and the expected challenge of each experiment. We haven't really looked into all that, but that will show up at some time in the results. And we aim for kind of like a naturalistic measure of replicability. So labs were told to follow the published protocol, but inevitably the published protocol has gaps, right? So we get like stuff like biopsy mice were housed in pathogen-free conditions and fed food and water, and you don't know the age of the mice, the sex of the mice, the weight of the mice, etc. So we actually sent out the protocols to each lab with all those gaps, and labs were instructed to fill those gaps as best as possible. They did not get help from the original authors. We did contact them, but we did not use this in actually developing the protocol, and each lab was free to take their decisions on how to best fill those gaps. They're also free to eventually perform necessary adaptations, which they thought was vital in the original protocol. So, I mean, actually, pretty much we told 3 labs, do your best possible replication, but this is kind of an ecological measure of 3 labs doing their best to replicate something based on what's written in the original article. And I think this is healthy in terms that you actually get naturalistic real inter-lab variability. We're not over-standardizing on any particular process that could lead us away from the original experiment. We did follow a confirmatory mindset, as most replication projects do. So every protocol got internally peer-reviewed usually by another lab in the initiative that was not performing this experiment, and by the coordinating team. These protocols were pre-registered at the Open Science Framework. Data collection was done via standardized forms. We did this very detailed Excel spreadsheets with all the information that we wanted to be able to audit the experiment, and we did pre-register an analysis plan for the whole project. That said, I mean, you can do as much quality control at the protocol level as you want. People still break their protocols. So as we received the experiments, we realized we had to adjudicate that in some way, because there's going to be minor breaks that are inevitable, but there's going to be like major breaks that may invalidate a replication. So after we got all the data collection forms, they were reviewed by the coordinating team. We listed all the protocol deviations and all the issues that came up in each experiment, in each replication, and sent this out to a validation committee that was formed by the coordinating team and by labs working with each particular method. 3 people looked at this list of deviations, and if anybody raised concerns that this may invalidate the experiment as a direct replication, we discussed this live and decided whether to include the experiment or not. Importantly, this was then blinded to the results, so people got a log of the methods and everything that eventually deviated from the protocol, but they did not have access to the results in order not to bias this decision. And when everything works, you end up with this. So we end up with 3 replications of an experiment. Sometimes it's 2, sometimes it's 1. And we can do a meta-analysis of them to have a best estimate of the replication effect size. We have the original effect size. We can compare this, and based on this, we end up with 5 binary replication criteria, whether the original estimate is within the 95% prediction interval of the meta-analysis, whether the meta-analysis estimate is within the 95% confidence interval of the original result, whether the meta-analysis estimate is significant at p less than 0.005, whether most at least half of replications are significant in the same direction as the original, and whether at least half of the labs consider the results subjectively replicated. So this is a rare case in which all of them are successful. So 3 labs pretty much agree among themselves. So the estimate is very, very close to the original result. But this is the absolute exception. It's more usual to get this. I mean, they have this original result to this side. Our labs, if anything, get a result in the other direction, but it's not significant. So this one does not fulfill any of the replication criteria. But it's not uncommon to have this. So this is a case in which our 3 labs disagree completely. So one of them gets an effect that's like even bigger than the original one. The other gets an effect in the other direction. The other gets nothing. And in this case, you get a mixed bag of criteria, of course, because in this case, prediction intervals are huge. Pretty much anything fits. So like any experiment, pretty much any effect size will be replicated because our variability is so large that it includes most possibilities. In this case, accidentally, the estimates actually end up kind of close. So this is fulfilled as well. But of course, the replications, the meta-analysis is not significant here. Most replications are not significant, and most labs do not consider the results successfully replicated. So you can get this mixed results. Some project numbers in the end. So this cost around 1.2 million reais. That's around $200,000. It's big money for Brazilian standards. Not that much for, I mean, by the US standards. I mean, definitely our PCB costs much more than this. It took us 7 years of work. We expected the experiments to finish in 2021. It ended in 2023, mostly because of COVID. Really, I mean, labs closed for a very long period of time. When they got back to the project, they had like a million other priorities, and it was very slow to come by. I think like first experiment came out in 2020, the last one came down in 2023 because we had to close and end. And we actually spent a lot more time data checking and analyzing than we expected. So like our preprint came out early this year. We have over 200 authors. We made almost 1,000 purchases of equipment reagents. We have at the moment almost 400 meetings of the coordinating team, which is the one that impresses me most. 18 general meetings. We have some gigabytes of raw data, but this is mostly videos. So actually, if you take it, the process data itself is not large. We do have a lot of code to analyze because it's just because it comes from a lot of places and in a lot of ways, and we had to do a lot of harmonizing for this to work. We do have a lot of methods because there were just so many steps to the project. And this is where we ended up. So we started out with 60 experiments, planning 3 replications of each. So that's 180 replications. And for 143 of them, we get at least 1 data point in each group, which is not bad. So like we have like something like an 80% starting rate at least. So like some experiments got canceled because labs left because they couldn't finish on time or couldn't start on time, or because we couldn't get materials for a few experiments, mostly because of licenses. But we do get replications. Most replications got at least some data. But when you get to the validation stage, we lost a lot of them. So out of 143, 46 replications got excluded from the primary analysis during the validation process because 18 deviated from the protocol. 12 had an experimental unit that did not carry enough biological variability. That's mostly for cell line experiments. 7 had an insufficient sample size. 5 had documentation we could not trust. 2 had non-interpretable results, controls didn't work as planned. Some had more than one reason. So we end up 97 replications of 47 experiments in our primary analysis. So this means we do not have 3 replications for most experiments. Most have 2, some have 3, some have 1, some just did not get into a primary analysis at all. And so this is, after all this damage is done, this is what happens to the replication rates. So they vary between 15 and 45% for the 5 criteria. 45% of experiments are in the original, 45% of original experiments are in the replications prediction interval. Again, this is a very optimistic criterion because if we get a lot of variability between results, prediction intervals are very large. So this really gives the benefit of the doubt to the original experiment. 26% of replications are in the original 95% CI. 19% of replications are significant in the same direction. 15% of cases, we get at least half of replication significant. 40% of cases, at least half of labs consider the result as successfully replicated. That said, labs frequently use very loose criteria as well, such as, I mean, it just went in the same direction, irrespectively of statistics. So I think this is a bit optimistic as well. In terms of effect sizes, actually, if you take the whole sample, there is a strong parametric correlation, but that's because one experiment has a very large effect size and a very large effect size in replication as well. This is in log scale. If you take this guy out, it's an R of 0.2. which is nothing to write home about. If you take the whole sample, you have a experiment rho of 0.34, barely statistically significant. And of course, as expected, most of the replications have smaller effect sizes than in the originals. So the replications are light dots here. The original are the dark ones, and we do get a 60% decrease in the effect sizes expressed as ratios of means. And big caveat here, though, is we get a lot more variability than in the original experiments. So if you look at the coefficients of variation, they're around 2.5 times larger in the replication than in the original experiment. This mostly holds for in vitro experiments. If you take the animal experiments, they're kind of like in the same ballpark, although a bit larger. But if you take the MTT and PCR ones, this really makes a difference. So of course, this means that for many experiments, we ended up underpowered to detect the original relative difference because our standard deviations are so much larger that the standardized differences end up being usually low. So we actually did a posteriori power calculation to account for that. So we tried to see how much statistical power we had to detect the original relative difference using not the original standard deviation as we originally did, but using our own standard deviations. We did this via simulations, and you end up with a measure of a posteriori power for the original relative difference. Most experiments are still okay powered, mostly because we overreached. We had like 95% in each one, but a lot of them are not. So a lot of them have insufficient power. So this should probably not be used at least for the statistical significance criteria, right? And you can take this out and actually analyze only the ones in which you have better power. And in this case, the statistical criteria end up converging. So now we have 31% of experiments with the original in the replications PI, because of course we take out the experiments that have very large variability, our PIs get smaller, but 30% of experiments are statistically significant. So I mean, if you take the criteria tend to be now between 19 and 31%. I think that's a fair adjustment. You can look at other sets as well. You can just look at every experiment, irrespective of the validation process. This actually does not make that much of a difference, surprisingly, in the replication rates. You can be stricter and have only those with 2 copies, 3 copies, but then you end up with small samples, right? If you take the whole series of options, all combinations between criteria and analysis sets and other decisions such as statistical distributions, ways to handle PCR data, you can build a specification curve, do a multiverse analysis, and you end up with a median replication rate of 26% with an interquartile range of between 17 and 36%. So this is pretty much where I think things stand for us. We did an analysis of replication success predictors as well. In terms of predicting the t-value of the replication, interesting, the researcher predictions done via the prediction survey were the best predictors. If you actually take the question which they were asked to predict, which is whether you think the replication will be statistically significant, this is a 0.43 correlation between answers. Accuracy is not as large as in the psychology projects, but again, I think this is expected. I think it's harder to predict this obscure lab results and intuitive psychological measures. In terms of predicting effect size decrease from the original to the replication, then method, model, and the original coefficient of variation are the best predictors. So the animal experiments decrease more in effect size. The MTT experiments in cells decrease less. And the larger the original coefficient variation, the more it decreases, perhaps because they were more inflated to begin with. So this is the numerical results. I think where do we stand in terms of comparison with other replication projects? I think it's within the same ballpark. You have these pharma numbers from 10 years ago, more 13 now, saying 11%, 21%. Tim's project has RPCB has 40% of positive effects replicating according to most criteria. All of our effects are positive. You do need a statistically significant result to calculate power. And it's hard to compare, right? Because the samples are so much different. We have this quasi-random sample of a literature. The RPCB has like very high cited papers. The mix of experiments and methods are very different. There's Brazil versus Rhodes. I don't think we can get into the comparison of why numbers differ exactly. But again, I think they're in the same low range of less than half of experiments replicating, which I think holds for every systematic replication attempt in lab biology, as far as I know. But we can go beyond the numbers, and I think most of the learning here goes beyond the numbers. I mean, about the literature, all we can say really is like, yeah, we could not replicate this finding. Maybe 3 labs could not replicate it. Maybe it does not look robust, but we cannot really know what happened. I mean, what's the problem with the original result if there is one? But we actually do know very, very well what were the problems in our applications. When things went wrong, when people broke the protocol, when they could not get the method to work, we actually have a very good log of what happened. So at some point when we started getting results back, I think we realized that the richer account of replication problems in the literature is actually our own experiments rather than the literature. When we realized that, we started a self-assessment project with the labs. We asked them if they agreed with validation, we made a decision, especially, and most of them agreed, even when we invalidated the replication. So I mean, it's not like we're in frank disagreement with the labs. If you deviated from the protocol, why did this happen? Could this have been prevented? If you were to start over, what would you do? And I think a lot of the learning from the project actually comes from this assessment. So if you look at protocol deviations, for example, they do have multiple causes. You can group them into categories: experimental issues, logistics, choice, lab errors, our own errors. And most of them are really issues with the experimental model that sometimes cannot be anticipated, right? So, I mean, you can try to build your very detailed protocol and go for it, but in the end, I mean, the cells don't grow this way, and you end up having to adapt, having to do something different. This was very common. So like, yeah, we planned to grow the cell for 2 days, but it didn't really work. So I had to put this extra supplement and grow them for 6 days. Otherwise, you can't even start. This is very frequent in lab biology. I think this has to be taken into account when trying to like pre-register methods from scratch. Perhaps you do need bio experiments before you do that. A lot of the protocol deviations came down to things going wrong in the road generically as well. So I think in Brazil, I think things go wrong frequently in terms of logistic, bureaucracy, supply problems. So like sometimes, yeah, the supplier ends up delaying the delivery of reagent X, and then reagent Y expires, and you have to use it slightly expired, or the animal facility could not provide the animals in the age that I wanted, or the equipment broke here and I had to use something that's slightly different. We're not very robust to these contingencies. I think we depend on weak links that sometimes break, and this leads to a lot of improvisation. And of course, but there's also human issues, right? And I think a lot of the room for improvement is here. I think some labs just like registered something in the protocol and did not really feel committed to this and ended up doing something else just because they felt that the pre-registration was kind of like this loose restriction of methods that were not to be followed exactly. Sometimes people just don't plan well and end up not reading the protocol in detail, messing up in something. And if you actually ask labs what would they have done differently, you usually get like better planning. Yeah, we have revised the protocol in detail, we have reevaluated and asked for the right quantity of reagents, we have organized the lab better to have more people doing it, etc. And I think from this come a lot of, to me, interesting takeaways on replication, actually on confirmatory science in general, within lab biology. One, I think there are real limits to pre-registration and to the concept of direct replication, like we'll follow the protocol exactly. I mean, sometimes following the protocol exactly leads the model not to work. And you do have to adapt because complex methods sometimes work differently in different labs, and it's sometimes impossible to follow the lab to the protocol strictly. Or I mean, yeah, we grew the yes. If the methods say, yeah, we grew the cells for 1, 3, and 5 days and got 50%, 80%, and 100% confluence, nothing guarantees that you get the same. You might do 5 days and get 60%, and then you either have to extend the days or use lesser confluence. So I mean, there's a lot of small decisions of what's the most direct replication here, and it's not always just following the protocol strictly. I think the best attempt at replication sometimes does involve tweaking with the method until you get something, the model working as you want. And this means it's actually pre-registering without pilot experiments, as we tried to do, because we had to have the protocol to like buy stuff and whatever. It's really kind of like sometimes not really viable. Second one is, I think we lack terminology for very basic things, and it's sometimes hard to uh, speak the same language in lab. I mean, labs do have very established practices and traditions of particular things, and sometimes they have done things this way for a long time, and they can even really describe what they're doing. To them, this is just the obvious way to do it. but the other lab does it in another way. The most obvious example is the experimental unit in cell line experiments. So what's your N of 1? Is it a different passage of the same culture? Are you maintaining cultures in parallel? Are you getting one culture and like doing subcultures within a plate and doing the experiment there? Are you pulling different cultures? I mean, people have very different understandings of this. Some are wrong, some are right, but I mean, many are valid, but some carry more biological variability, some carry less. I think in terms of interpreting the results, this makes a difference. This is very rarely understandable from the methods. And this was actually very rare. I mean, we thought we made it clear in the protocols. We had like, yeah, use different passages of the cell line, so do the experiment in different days. People interpreted this in like 4 or 5 different ways. And we had a hard time even understanding what they did. I mean, sometimes we had to exchange like 3 or 4 emails just for people to explain because there's no common language to talk about this. And I think this actually is a simple thing that could be improved in terms of making cell line experiments more reproducible. I think it holds for other aspects, for particular types of controls, of blanks. People do use different terminology. I think standardizing some things would help. A third one is something we anticipated. I think this is a big problem in lab biology. So if methods are hard, if it's hard to, I mean, getting the method set up is hard, you run into a lot of methodological failures, right? I mean, sometimes the model just doesn't work and you don't really get a result. It's not like we did not replicate the result. You can even judge the result. And then there's results that don't replicate. And I think people conflate this very frequently. And I think this is actually a big problem in biology because, of course, the methodological failures get thrown into the same this didn't work bucket as those experiments in which you do not get the results you want. So people just, yeah, let's do it again because it didn't work, and you do it again, and eventually get the results you want. I think this actually explains a lot of the problem in biology, but this is just a personal perspective. We were aware of this. We tried to clear this up front by having people set up explicit methodological validity criteria. So if I get this, and this working in the method, then I had to consider the result. This was a failure. I mean, people had a very hard time in establishing this a priori. Labs did not understand the concept. They frequently set up impossible criteria. So yeah, we only use RNA that's like 95% pure, but then you get like 92%. And of course, people use it because, yeah, this is good enough. So like people had a very hard time doing this, and I do not think this worked in your average academic lab. The a posteriori validation, on the other hand, I think was a good call here. We made that up in the middle of the project, but I think that worked. I think like actually having access to a methodological log of the experiment, whatever went wrong, without access to the results, was a patch that allowed us to differentiate these two things. And I think this is much more feasible for your average academic lab. I think this is a practice that if I still had a lab, I'll probably implement. Uh, and then there's the human aspect of it and the cultural aspect of it. So like I think one thing we realize, and I think other people realize as well, I mean, people in biology do not have a very confirmatory research mentality. People are not used to this. They're not used to pre-registration. A lot of people just wrote generic descriptions rather than commitments to be followed. Labs have very established ways of working. Standardizing practices across them is difficult, especially when we're like not really paying people directly. And definitely, I mean, there's a lot of improvement in terms of data management, documentation. We did get very messy data sometimes and like silly errors in terms of inconsistency. We spent like I think 6 months just doing data cleaning and checking with the labs looking for inconsistencies. And again, people are not used to working in this coordinated fashion, right? This is review 2023 review of preclinical multicenter experiments. They looked at like all of PubMed, found 16 results, which means, I mean, people in biology are not used to doing the same thing in multiple places, and this makes it very hard to coordinate. So I mean, not only people are not used to being coordinated, which is an issue, we're not trained for this as well. I was never trained for this, and communicating with like 200 people is very hard. You need a lot of hierarchical coordination, and like so it had like me as coordinator and like 3 coordinators, one for each match, would like talk to people in the lab. So it ends up a complicated structure. And I think we clearly underestimated the complexity of just like getting data from different places and having to check, clean, harmonize, analyze. It turned out to be much more complicated than we expected. We put a lot of quality control investment in the protocol in the end. Like a lot of things happen after the protocol which do not follow it. And I think we also underestimated that, I mean, the need for systematic continuous quality control. We did not vet the labs in the start. We could not, I mean, we could not find a good way to do this. And I think these things have to be cleared up for and be done more professionally for these projects to thrive. I mean, we did learn a lot the hard way, which is like doing stuff and getting hitting our heads against the wall. But if we really want to make this confirmatory science more of a more day-to-day thing in biology, I think we do need specialized people to run this. And really, my best quote about the project is really, I mean, we thought we'd just like get a lot of people together and merely doing this, like do this very rigorous confirmatory science, but really it's like you can put a lot of garage bands together that does not make an orchestra. And I have nothing against garage bands. Here's me playing in one. And I think I mean, exploratory science is healthy, is important, is how most of biology goes, and perhaps this should be the way. But there's also need for orchestras, right? I mean, even if most labs are still doing exploratory research their own ways in small groups, that may be fine. But at some time you have to confirm a few findings, and when we want to get into to that direction, I don't think we have many people, or we have people at all that are used to working in the orchestra mode in most fields of biology, because just the field is so much very skewed towards exploration. And again, I think both garage bands and orchestras, both exploratory and confirmatory science, are important. I think we're definitely underfunded, under structured, under everything for confirmation. And really, I think this actually does not, it goes beyond cultural change. I mean, it's not only like changing the minds of individual researchers, it does involve a restructuring of academic labors of institutions for this large consortia or for specialized institutions to be able to do this kind of confirmatory science, to have people trained in managing it, funded to manage it, and trained actually working in more collaborative fashion. I mean, we do collaborate a lot in terms of like, I do my thing, you do your thing, and we get the data together, publish a paper. But like, let's try to do the same thing more or less and standardized procedures. Not super common, and I think we do need a little bit more of this, but this actually requires reinventing a bit of the way we work within academia. And I think we have wrote about it. A lot of people have written about we should be more creative in terms of thinking of different ways to do research and organize people. I think COS is actually one of these attempts, and we're very happy to be here because of this as well. And of course, our main spin-offs here are attempts to get researchers collaborating in different ways. We have BRISA, which is a collaboration for Brazilian researchers doing systematic reviews to actually learn about the methods together and do some collaborative work. And we do leave the Brazilian Reproducibility Network, which is our main, should carry forward whatever we started building, which is, as other reproducibility networks, an organization that brings together groups, institutions, people, journals, societies, to talk about reproducibility and how to improve it in the academic environment and outside of it as well. And this is it. I'd like to thank my coordinating team. Part of it is here with me today. Thanks US for the opportunity. Thank our funder, the Happy Data Institute. But fundamentally, thank the over 200 researchers around Brazil that allowed us this very privileged view of real life labs working in real time. I mean, we learned a lot from these people, and we're very grateful for the collaboration. Our preprint is out. We have a lot of data out of as well, if you want to take a look, and we're always available to discuss it if you think it might be valuable for you. Thank you.
That was excellent. Thank you so much for that. Really, really appreciate that overview and all the insights that are there. So just a reminder to everyone that's on the webinar, if you have questions, and I already see some, pop them in the Q&A. I'll help navigate this with our attendees here to go through them. I'm gonna steal and ask one first before we kind of dive into some of the questions that are there. And if it's okay, I'm gonna go back to the title of the project in your talk, which is it's a Brazilian biomedical research, right? It's on the Reproducibility Initiative for Brazilian research produced. I'm just curious from your perspective, if anything, that this says about Brazilian research, and really to me the bigger one is what as an international research community should we take away from the project? You gave, I think, a great overview of what you found, the challenges, the opportunities, and how to go forward. But I'm just kind of curious to go back to the fact that it was, from the beginning, very focused on the research occurring in Brazil.
Yeah, I don't think you can make any comparisons. I mean, if you restrict yourself to Brazil, I mean, we can speak about this reality, I don't think it's that much different, but of course, I mean, whatever I think is just a guess, right? I think some contingencies are worse here in terms of unpredictability of a few things. So I think we do improvise more. I think Brazilian researchers are usually proud that well, yeah, we do a lot with so little, and I mean, we're able to get around this hurdles. But I think improvisation at some time, I mean, it has its costs. So I think, but I have no idea. I think a lot of improvisation is just because people are just very used to doing research in an exploratory fashion. I think my scant experience working elsewhere does not suggest that it's very different from elsewhere. I hope most of our takeaways are actually useful for lab biology as a whole. But of course, I mean, I can only speak about our data. Not sure if Clarissa or Mariana have something to add.
Okay, thanks. I just wanted to put you on the spot just for that, just because the name of it. I completely agree with you. There's so many similarities to research that was not done in different contexts from projects we've done. I very much agree with your overall assessment. All right, so I'm going to go through and help direct. I'm going to go through the first question, if it's all right. That's actually was over in chat, and you kind of hinted at this, so I'll read it and let one of you guys answer it. Someone wanted to ask about learning about the role of research support staff. They're very interested to learn about the contribution of support staff to reproducibility, knowing if those individuals were engaged in the work that you were doing. If not, even just what your thoughts are in general on that topic.
Yeah, so we took a very loose approach in terms of like how labs organize themselves. I mean, if you consider our team as support staff, I think I mean Clarissa, for example, was pretty much overlooking every stream of the elevated plasmids. For example, I mean, we were acting as distant support staff. So we helped with protocols with like I mean people did call us up in the phone to like, yeah, this didn't work, what should I do? But we tried to leave decisions to the labs. We made the labs very independent, and I mean this was just like our naturalistic way of doing things. We were, I think, I mean, we weren't. I wasn't anybody's boss there, so like, it's grown-up adults running their own labs. It's hard to say, yeah, do this way, and so like teams organize themselves in their own way. Some people use more research support staff. Some people use less, and I don't think we have a good log of that. And and again, I think if I was to go back, I would have perhaps had more information on how, like, asked for more information on how teams were actually organized. I think this would have been interested. We took this loose approach. Yeah, just indicate three people that will be working there. And I mean, we don't really want to know who's doing what. I mean, organize yourselves. This actually ended up being a problem sometimes for authorship because, like, in the end, if sometimes says, yeah, people X, like person X left the group, but like person X actually said, no, I did something, we could not adjudicate. So like perhaps this would have, I mean, I would have gone for a slightly more hierarchical coordination, less democratic, but like have somebody who's responsible for, yeah, I'm responsible for saying who did what in the team. We did not have that. I think this would have been useful. We do have contribution who contributed to each experiment in terms of protocol and data collection, but I would have done a more complete job of trying to see how labs organize themselves. I really do not have Clarissa. Not sure if you actually have a better view of that as you're in closer contact with the labs.
Yeah, I think what I can add to that is that more junior personnel in the lab were more directly responsible for executing the experiments. And yeah, so most of these junior folks were postgraduate students, but sometimes it also included like technicians in the labs. Yeah, but again, like Olavo said, I don't really know the exact role and responsibilities each of these people took. It's just yeah, I because of the contact I was having with them, I know they, yeah, sometimes or most times they were the ones running things and communicating with us.
And this might be something actually slightly unique to Brazil. I think like Brazil has a very junior scientific workforce. A lot of the work is done by graduate students. Postdocs are relatively rare. Undergrad students actually take a very active role in research done at universities. So I think, yeah, and I think that also has its costs sometimes. I mean, sometimes execution of the experiments get to be done by a very junior person. And I think we did have examples when much this may have explained something that went wrong.
Thanks. No, that's great. Yeah, it's always important to keep in mind all the different contexts, both country, but also just research intense universities versus some that are still doing great research, but just not with the same resources. All right, there's another question. I'm going to pivot to this. This is a great one. So, just kind of pushing on that taxonomy for standardization, the idea of taxonomy for standardization of language and protocols. Somebody was asking, is that similar to a dictionary for data management? So just kind of your thoughts on, and I think this is a good one that just if I can expand to is the idea of it's like a recommendation, right? This idea of both the original and the replications, right? It's just a chance to say, how do we do a better job? What are your thoughts on that if you just want to expand a little bit?
Yeah, so I like so for the cell line experiment unit I mentioned. I think you can describe like what is your unit in like four or like at a point we're just like so frustrated like not understanding things. We actually built a spreadsheet and had like four columns. Like, are experiments done in the same day? Are these passages of the same culture, or these cultures run in parallel? Are these so like in 3 or 4 letters, like yes or no, we could. So I think it's you can just like create and yeah, I mean create a standard code. Shouldn't be different. I think sometimes taxonomies can be really easy. I come from the medical field. Like sometimes, like if you take just people were reading mammograms and describing them with words with very complicated, like yeah, this is a nodule that has a few things like this and that. A lot of text. And sometimes some people just got fed up with this and like created BIRADS, and I was like BIRADS 1, 2, 3, 4, 5, which really encapsulates, do you need to biopsy? I mean, do you need to follow it more closely? Do you need to biopsy? Do you think it's cancer? And which are the things that matter? So I think perhaps extracting our methodological descriptions are very arcane, sometimes they're very textual, and sometimes it's just like, yeah, we want to hear about this and this and this, and these are the points you might work. That said, I think the experience we have with like reporting guidelines in lab biology especially is mixed. So I mean, I think some reporting guidelines worked very much in the clinical field, such as Consort, Prisma, etc. Arrive has had limited uptake in lab biology. I think there's clinical trials are saying that even if you include a checklist, this doesn't really make a difference. I think other guidelines, the Mickey guidelines for PCR, exist, but people, I mean, they're not widely followed. So it has been a challenge to implement this. I think there's a room for standardized taxonomies, but it do not. Implementation is not trivial, and I think a lot of efforts in lab biology have actually not worked as planned in this field. So I am a fan of the concept. For implementation, I think it's a harder thing to, but I think there's ways to do it.
That's great. Yeah, I think you're right in terms of if this is a chance to evolve. So let me ask another question. It's a good one, right? So Philip is saying the general findings are quite discouraging when we think of, right, you're looking at state of the art. This is research, new research. And it can lead to questions if there's how much credibility in the published scientific experimental results we should have. So it's a good question. Here it is. If you are a funder that would give you confidence that funding, sorry, if you are a funder that would give you confidence that funding will be actually a result in advancing our understanding of the particular area of biology the funder is interested in. So I think he's asking here, what do you take away, right? If I'm a funder trying to invest in this research, like what is based on these results, kind of what do you think about in terms of the confidence you should have in funding specific areas?
Yeah. One general comment is I think like what we did is something that funders should be doing. I mean, to me, this basic quality control, I mean, just let's replicate a bunch of findings and see how easy it is something that to me would be in the general interest of funders. I mean, this was something I pitched to Brazilian funders. Yeah, this. I mean, we do need these numbers, and we just embarked this because like none of them would actually do it. And so I think investing a bit in this client, I mean, it's a bit expensive, but I mean, in the end, for like the whole budget of a funder, even in Brazil, this is like our project is not that large, right? So I think funders can try to do this, and I do think funders should invest in confirming some findings. I mean, if you want to advance an area of biology, I think one thing we would lack in doing is like try to identify the key things that we really want to confirm in a very rigorous fashion, right? I mean, I used to work in the neuroscience field, and like people argue for like 20 years over whether a given phenomenon exists. Because person X can get it, person Y cannot get it, and I mean, they get different results, and they try to resolve this in the but it's very slow, right? I mean, each one is actually writing his own papers in his own labs, and I mean, if we just like got the whole field together and like, oh, let's do this thing in 10 different labs and like try to figure out the controversy, we could do it, right? So I think this coordination project is something that can be done, and I think perhaps funders should be invested. I mean, every year, so if you want to advance in area of biology, try to, I mean, every year identify 2 or 3 or 5 key findings that you actually want confirmed and like fund specific research to confirm this. I think this is actually a good call. Of course, like what are the important findings is an open issue in which the community should pitch. But I think learned societies, for example, could pitch in this direction. So like if the Society for whatever neurochemistry in every year tries to highlight like, oh, these 5 developments are important and this deserve to be confirmed, perhaps we can actually like fund a specific specific projects that actually try to be confirmatory projects. It doesn't have to be the whole budget, but like perhaps like a small part of the budget could go towards confirmation of what's out there. And I think of course you cannot replicate everything, but I mean this would at least strengthen the main things that we want to see confirmed, right? So fund a few orchestras in the end, I think is and select what the orchestra are going to play. I mean, we have to figure out what's worth confirming, which we did not do. We did this very random sample because it's kind of like a meta research project that's interested in reproducibility rather than in the actual findings. But I mean, if you're actually interested in confirming the findings, I think actually picking up what to confirm and how to confirm it is a big part of it, and it does require deep expertise, right?
Right. No, absolutely. This actually threads well to a question. You can probably read it, and Plant has it. There's a lot of context background, so I'll let you read that. But I like this question. It's a play off of what you were just answering, which is right. There's a lot of sources of variability. It does take a lot of work. Definitely a change in mindset. It'd be great if people were thinking more about sources of variability, and really how to kind of better understand kind of what are the specific predictors of specific findings and how to generalize those. And then that's confronted on the funder side, right? Which is we only have so much resources at any given point to devote towards research. How much of that should be spent on confirmation replicability studies? So I think there's a trade-off here, and the question is, would you agree, and what are your thoughts on getting to consensus knowledge over time with many labs using many different approaches, so triangulation? Is that still reasonable for biomedical research? I think you can expand on it to what you just said, which is, you wouldn't replicate everything, you'd replicate a subset. So I don't know if you want to like take Anne's question about how does this fit into triangulation as still being a way to kind of accumulate knowledge and sources of variability over time.
Yeah. So I mean, we did not triangulate. We took an intermediate way here, right? I mean, we did not super standardize things. We let each lab do their own methodological decisions. Like, there's protocol gaps, you fill yourselves. Like each lab was free to adapt the protocol as they saw fit. Although we still made the orientation is still try to perform as as much of a direct replication as you can. So I mean, we're not picking up some different method. I mean, try to stick to the original one. But if you want to adapt, if you want to fill in gaps, I mean, you do it independently, which I think was our way to try to avoid over standardization. Which I think is a real problem. If everybody starts doing this method in this particular way, you can end up standardizing to a way that is actually fails to pick up some phenomena, and I think diversity is good. But of course, I mean, on the other side of the spectrum, you can just say so loose that like what actually ends up happening very frequently in biology is like people are always nobody's working with exactly the same method. So like I get result X, you get result Y, and there's always this little difference that people attribute result. I use mice, you use rats, or you, and then knowledge does not accumulate because like there's always this hidden moderator that you can invoke to say, this is why we get different results. So I still get result X, you still get result Y, and we still keep publishing for 10 years, and nobody really resolves what's. So I think there's a middle of the way here, right? I mean, I do feel that I think your paper in PLOS Biology, I think, speaks to that as well. I mean, there's no real split between direct and conceptual replication. I mean, things diverge slowly. And you do, I mean, doing something completely identical is probably not super informative, but doing something that you can always invoke a hidden moderator and say, yeah, this is different because of that, does not constitute a replication anymore, because you can always, I mean, it does not make you lose confidence in the original result. So I think you do need replications that are ideally diverse enough that you're actually looking at robustness and generalizability, but not that different that you still think this is evidence against or in favor of the original result. The thing is that that's very hard to encapsulate, or there's no way to do statistics about what's close, what's different. I mean, that's like, what is evidence against the original result? How much should I distrust the original because of this? Is a super tacit question. Like, I mean, there is some kind of Bayesian updating, but I mean, people will vastly diverge in like, yeah, this is, yeah, how close this is. I mean, is this still a valid replication? There's no easy answers there, and that's why we set up a validation committee with like 3 people who are doing their subjective thing. Oh, yeah, this is like, yeah, if you were to incubate this for 30 minutes and you did it for 32, is okay, but like maybe 43 is not. This is a completely, yeah, I don't think we have that coded. So I think there's a lot of subjectiveness in how much yeah, how do you bring evidence from different methods together. We don't have statistics for that, and that's complicated. So I do think there's value in trying to stick to the same question and agreeing up front that we're sticking to the same question, because at least, okay, if you agree up front that this is kind of like a replication, we can still use this as evidence, synthesize this, and like get to a best answer. The more things diverge, the harder it is to agree on what's diagnostic of what, right?
Oh, that's great. This actually feeds into the next question very well. I'm going to read this one out, mostly because I want to say the first 2 words, because I completely agree with it, which is amazing job. But can you elaborate on the point of how a real replication comes from not following to the letter of a pre-specified protocol? You were just saying it now, actually, right? Which is there's some flexibility because of the fact that we don't have the tools yet to even specify it correctly. I completely agree with that. But then how does that reflect on your results, right? So since, and again, he's, I think, a valid understanding, replications are valid if they don't deviate, or they can deviate, but within what bounds, right? So how would you see the replication projects like this working if they were to incorporate this flexibility in their proposal? So I think that's kind of what you were just getting at, but let's confront that with the process of pre-registration, right? You're saying specify it, yet you can deviate. I think maybe you want to just push a little bit more on how this works in the context of wanting to still do replications, yet embracing this flexibility that you were just talking about.
Yeah. So again, I think really we're incorporating, like, our definition of replication is incorporating the subjective beliefs of people who are doing the replication and of people who are validating the replication. Again, I think there is non pre-specified subjective calls that, like, yeah, whatever, 35 minutes, okay, 38 is not okay. It's in somebody's head. I think people will diverge, and I don't think so. Our results definitely incorporate this in terms of like defining replication. I think this is a collective construction of the people who built the protocol, with the people who reviewed the protocol, the people who work in the validation committee. I think their beliefs end up deciding this. I think this is okay, and that's why we had a lot of people, because people will not necessarily agree on that. On how to design this replication projects, this is a good question. So I think our project is very much like a one-shot replication project. Like, do your protocol up front, try to follow it. We did not really leave that much room to adjust afterwards. And I think those will lead to different numbers. So like, if you just like do your one-shot replication, which have to specify everything in advance and hope this works, I think your replication rate is likely going to be lower than if you can tweak the protocol here, and try to get as close as possible to the original results in multiple attempts. Of course, in the limit, this also, I mean, if you just like can try, eventually we get the same result just by insistence. So I mean, you can overestimate replicability if you just like are free to try as many times as you want. But perhaps, I mean, what's the fairest concept of replication? I'm not sure. It's somewhere in between. Perhaps it's I'll give you the protocol, play around with it, try this and this and this, and then pre-register your methods and then do one shot following your best shot pre-registration after a lot of tweaking with the protocol could be perhaps the fairest approach. It is complicated to do. I mean, it would be very complicated to organize because you have to like give a lot of reagents and then people like change their minds and like buy them again. So in a systematic way, it's very hard to organize. But I do think that's, I think, closer to what labs do in reality. I mean, if labs are trying to replicate something from the literature to build up on, they will not stop at the first attempt. They will try 1 or 2 or 3 or 4 times. But of course, I don't think the pre-registration, like, yeah, okay, now this is what's working. Let's do my one pre-register experiment step actually happens. So perhaps that's the one I should add, because otherwise it's just like it ends up being a lot of confirmation bias. People just try to get these results that came up in nature. Sometimes eventually somebody will get it, right? So I mean, everything ends up confirmed. If you insist enough, there's like 10 different labs trying 2 or 3 or 4 times. Eventually somebody will get it and we end up confirming everything. So
You want to say something, Mariana? Go ahead.
Yeah, just to add a point that usually when people do that in the lab, they do lots of times, but they know the results, no? They know the number they want to reach. So the idea would be having it, but not having access to the results in all these tryings. So maybe it would be a little bit more adjusting the methodology and not just trying to reach a number. But anyway, it would be many tries, and then the chance to replicate would be better, not only for methodological adjustments, but also for trying many times. But
and this gets back to conflating methodology and results, right? I mean, if people are judging, I mean, people will judge that the most appropriate replication is the one that got the closest results. This is like a bias that almost everybody has. And if you don't get the same results and you think that something was wrong with the methods, I think you also had that in RPCB. So I think really making people evaluate this separately is really valuable, and I think it's something that people underdo in the biology as a whole.
Yeah, I completely agree. I completely agree with you. All right, we got 2 minutes left. So I'm gonna give you one more question. You're gonna have to give it like a quick response. I know this is a good one too, right? So from your results, you had the best predictor of replication was the scientist's predictions. That is very interesting. So the question is just, do you support using prediction markets such as part of the grant review process of experiments? Like, do you think this is a tool?
Perhaps. We have not looked into that in detail. We do have justifications for the prediction, so people actually answered why did they predict X or Y or Z. We have began to classify those answers, but we haven't really looked at it. So I'm very curious to know why this worked. I'm not sure what people are taking into account. As an anecdote, if you actually just take a look at like this general rigor measures like blinding, randomization that people usually use to assess animal experiments, this actually correlates inversely with reproducibility. So like the more bias control measures in the original papers, the less reproducible it was. I mean, it was a small sample because it's just animal experiments. It's barely significant. But so it doesn't seem to be that. I'm not sure what people are looking at, effect sizes. So I'll be curious to know. We haven't really, we're probably going to have a paper just on prediction in the future, but I think it's important to, there's probably something there. I mean, it doesn't seem to be an accident that people were actually able to predict something, but I'd be curious to know what they're using, right?
Right. Good. It's like all research, it doesn't stop. I love it. Yeah, everyone take a peek at the paper. It's out. Definitely reach out. I just want to give one more big thank you. Thank you for the work you did, and thank you for taking the time to talk with us today. It was really, really a pleasure.
Yeah, thanks a lot. Thanks for everybody for being here, and I mean, hope to see you around. And feel free to get in touch. I think my email was in the slides. I can actually send it in the chat to everybody, or maybe I can't, but yeah, I can. So just paste a few links here. But I'm Olavo at and whatever. The slides are. Oh, now I lost it. But and I'll post a link to the slides here. It should have every other link there.
Perfect.
You probably find it by archive.
Perfect. Awesome. And thanks everyone for attending too. Yeah, we'll have this posted so that you can share this too with your network when that happens. Awesome. Thanks everyone. Have a great day.
Thank you.